Okay, good day everyone. Um, this morning we are going to be discussing about the origin and history of light microscopy. Uh, the conceptual origin is that is actually the discovery of lenses. The history of light microscopy begins with actually discovering curved lens that can magnify an object. And long before microscopes existed, ancient civilizations such as the Greek and Romans observed that glass spheres filled with water could actually enlarge images. All right. So, however, practical lens making only developed properly, I think, in the Middle Ages or so. And by the 13th century, spectacles were being produced in Italy. Now, this was the technological foundation upon which microscopy would later develop. Okay, so the invention of the microscope required two essential development. Number one is high quality glass production, and number two is understanding of lens curvature and focal length. Now, the first compound microscope was developed in late 16th to early 17th century, thereabout. So, the first true microscope appeared in the Netherlands around 1590 or so. And so, the um, the invention is commonly attributed to Zacharias Janssen, who is a Dutch spectacle maker and possibly his father Janssen. They reportedly, what happened was that they reportedly placed two lenses, in tube and observed magnification. Okay, but then this early instrument actually had poor resolution, okay, produce images that are distorted and had low magnification power. So they were scientific curiosity rather than they were just scientific curiosity rather than reliable research tools. Now let's talk about in the early 600 Galileo's contribution. Around that time, a man called Galileo improved the compound microscope design. He referred to it as Ocholino. That's the little I, Ocholino. Okay, so Galileo's version is that he used improved lens combination, provided better magnification, marked the transition from curiosity to scientific instruments. So while others were still curious, Galileo was the one who actually, marked that transition from curiosity to scientific instrument. Now, this period actually established microscopy as a legitimate observational tool. What is the role of Robert Hooke in the 1665? Okay, and Robert Hooke is attributed to the cell theory, right? So a major milestone occurred around this time when Hooke published his famous book called the Micrographia. So using a compound microscope, he was able to examine thin slices of cork and observe small box-like compartments. He called them cells because they resembled monastery rooms. Okay, so this was revolutionary because it introduced the term cell, it initiated microscopic anatomy, and it laid the groundwork for histology. However, Hooke was observing dead cell walls, not living cell. Okay, so you have to note that. Now there's another person called Antonie van, I've forgotten the last name, Antonie van. We call him the father of microbiology in the 16700. Okay, that's where the real breakthrough in microscopy came from. Anthony was the real breakthrough. Unlike Hooke, Anthony used simple microscope with a single very well polished lens. Surprisingly, his microscopes were superior to many compound microscopes of his time. All right, so he achieved magnification up to 200 to 300 times magnification, which of course was extraordinary for that time. As the 17th century. If you ask me, he was the first to observe bacteria, protozoa, sperm cells, red blood cells, even muscle fibers. He was the first to observe them. So for the first time, humanity saw microscopic world of living organism. And this discovery actually completed a completely transformed biology, all right, and medicine. Now, in the 18th to early century, there was optical improvement. Microscopy advanced slowly during this period due to a major problem, and that was the chromatic aberration. That's the colored halos around images due to light refraction. Okay, so the solution came in the 18200 to 1830, so for 1820 to 1830, um, with improved lens design. A man called Joseph Jackson, developed a chromatic lens which now corrected chromatic aberration and greatly improved the clarity. Now, this period marked the transition to modern optical microscopy. Very important period. Okay. Now let's look at the contribution to cell theory. That's in the 1830s to I think 1840s or 1850. I can't remember. Improved microscopes. Actually, at around this time, improved microscope allows scientists to study tissue more accurately, and the key contributions are from people like I think Matthias Klendin, Rudolf Virchow. I think there's another one. His name was Schwann. Okay, that's where the Schwann cell was bettered. Okay, so they actually established the foundation of cell theory, which is number one, all living organisms are made up of cell. Number two, the cell is the basic unit of life. Number three, all cells arrive from pre-existing cells. Okay, this is the cell theory. Okay, and this actually was the it was a turning point in pathology and human genetics. Okay, very very important. Now, without light microscopy, there will be no histology, there will be no tumor diagnosis, there will be no understanding even of cellular inheritance. Okay, there's another man we didn't have not mentioned. Okay, I think it's in the late 19th century. A man called the Abbe, Abbe. Okay, of course he contributed to the science of resolution. You know, I think Ernst Abbe. He developed the mathematical theory of resolution. He introduced the concept of numerical aperture. He actually established the diffraction limit of light microscopy, and he worked with a man called Carl, leading to the industrial production of high quality microscopes. So his formula, that's Abbe's formula, showed that the maximum resolution of light microscopy is approximately 0.2 micrometers, that's 200 nanometer. And this limit actually explains why viruses cannot be visualized with a standard light microscope. Then in the 20th century, several innovations improved light microscopy. For example, phase contrast microscopy, which was developed by Fritz Zennig in the 1930s, allowed visualization of living stem cells. We also have fluorescent microscope, okay, which is actually essential in immunology and molecular genetics. The basics for immunofluorescence diagnostics. Okay, we also have confocal microscope, which produces optical sections. Okay, and allows three-dimensional reconstruction. Um, of course, which is critical in advanced cellular imaging. Okay, now what is the importance, the clinical and genetic relevance of what I'm talking about. Light microscopy is actually very important. Okay, in histopathology, in hematology, that's blood films, cytology, for example, pap smear, even in embryology and basic genetic research. Even in the era of molecular genetics, polymerase chain reaction, as the PCR and the genome sequencing, light microscopy actually remains the primary diagnostic tool in uh, most of this field. Even in many African medical school, teaching hospital, uh, you know, the standard light field microscopy is still the backbone of pathology and studying um, histology. So the summary of it is this. Okay, in the 13th century, there was spectacle development. In 1590, the first compound microscope was developed by Janssen. In 1665, Hooke coins the word cell. In 16700, uh, this other man discovered microorganism, forgotten his name. In the 1830s, achromatic lens was discovered by Lister. In 1838 to 1855, cell theory was established. In 1870, Abbe's resolution theory, and in the 20th century, phase contrast, fluorescence, and confocal microscope were developed. Okay, so I hope this helped to put light to light the microscopy. Okay, so have a nice day ahead. Thank you very much. Thank you very much.